Hello everyone, today we are going to discuss about electron transport chain. So this is the second type. So in the previous class we discussed about the non-cyclic one, which is does not have the cycles and it have the two photosystems. But here in the cyclic one, the only one photosystem, that is a P700, is present. So here NADP, if it does not undergo reduction, it does not accept the electrons. So the electrons will move backward. So and they are accepted by the plastoquinone. So plastoquinone along with the protons from the stroma, the electrons will be accepted. So this downward movement later they transfer to the photosystem 1, which results in the formation of the ATP molecules. So here if you see, plastoquinone will transport the electrons from the stroma as well as protons to the next carrier, that is the cytochrome BF. Cytochrome BF accept only electrons and the protons will be pumped into the lumen of the thylakoid like the non-cyclic, and later these electrons will be transferred to the plastocyanin and later to the photosystem 1 with P700. Along with the exciting electrons, these will also be excited, transferred to the PS2 and next ferredoxin. So this will be continuous. So this is the cyclic one. Later the protons which are pumped into the thylakoid, so it will synthesize the ATP. So this is the cyclic one, and here the main difference between non-cyclic and the cyclic here is photosystems. Here only one photosystem one is involved, but in the non-cyclic both are there. So next is the chemiosmosis. The chemiosmosis is nothing but a ATP synthesizing process. Okay, this is first proposed by Mitchell. Chemiosmosis is the movement of the ions to ATP by the movement of the protons across a membrane during the cellular respiration or the photosynthesis. Here we are discussing about the photosynthesis chemiosmosis, right? And this for this purpose ATP synthetase, next complex is the ATP synthetase, which is also present in the thylakoid membrane, and this synthetase enzyme will synthesize, and it consists of two regions, F0 and the F1. So if you see upper region plastocyan photosystems, cytochromes, photosystem 1 2 both are present, and below, so all the protons which are collected in the lumen are entering into the ATP synthetases. So F0 and F1 regions is there. So F0 is a proton channel and F1 is a catalytic region. If you see which is embedded in the thylakoid membrane, which is accepting the protons, is the F0, and which is below that is outer side, that is towards the stroma, which is present, that is the F1, which is the catalytic one. When the protons enter, it undergo catalytic conformational change and synthesize the ATP. F0 is the proton channel, we already discussed that. So this will be, the protons from the lumen will be accepted by that proton channel and released into the stroma, which are used in the dark reaction by the synthesis of ATP in the by AF1 particle. Thanks for watching. Please subscribe my channel if you like it. Okay, we continue in the next class.